food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning, or before bed, or after meals, or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. A little bit about what you do today. What do you do for work, and what does that involve?
Well, at the highest level, what I would like to do is to bring true preventive medicine to the society, and that is to essentially empower individuals to live the same life that people in the blue zones do, which is they live into their 80s and 90s very healthy. They have physical health, mental health, cognitive health, and immune health well into 80s and 90s. And nature has already figured out how to do that for some people, those that are born there. Sadly, we don't have those kinds of lives in modern society in the developed countries, but we have the science to figure out how nature has enabled this kind of a long health span. And then with testing and AI, enable individuals to live that kind of a life. That's at the highest level. And then how I have gone about doing that, well, that's a 15-year story. So I'll just give a summary that about 15 years ago, I basically changed my scientific career. I was a government scientist, and I was mostly working on the drug development and drug discovery. And I realized that I actually wanted to do this preventive medicine strategy. So I changed my scientific focus in 2010 and started developing a systems biology view of the human body. And a systems biology view means that there isn't just one factor that affects our health. And so there are people who are geneticists and they focus on just the genetic contributions. There are people who believe that the gut microbiome is the root cause of all diseases. There are people who think that heavy metals are the root cause of all diseases, and so on. But the real truth is that for a population, there are many, many factors. And even for individuals, there are likely many factors that contribute to our health and disease. And we really have to look at the entire human physiology. And what I realized back then was that we needed to have a very solid molecular test that profiles that physiology. And at that time, I decided that I was going to pursue RNA and not DNA or other molecules in the body as sort of the best molecule to characterize our physiology. And so today, for example, what we do with our stool, blood, and saliva test is that we look for over 100,000 different kinds of molecules that maintain our physiology. And so it took about 6 years as a government scientist to develop this foundational technology. And then in 2016, I left the government scientific position and we spun out Viome with Guru Banavar and Naveen Jain. And now, over the last 8.5 years, we've been running the technology at the company and scaling it and adding a lot of AI and adding a lot of knowledge and so on.
Mm-hmm. So when you talk about, so you spun things out and did this company based on the research you were doing at the government. You have an academic background, and you're talking about preventative medicine and systems biology. What sort of led you to that transition? Why did you not stay on the government research track you were on? Were you not able to do preventative medicine stuff in that context, and so you needed to switch out of it? Like, how did that sort of transition happen, and what was like propelling you?
Yeah, so when I initially envisioned this whole platform, I did not make a decision as to what kind of an entity was going to actually exploit this sort of a platform. And as an academic, my natural sort of tendency was, well, I'm just going to start a nonprofit institute, right?
Mm-hmm, mm-hmm.
But as years went on and I did more research and I did more talking, I realized that the only sustainable way that this can be brought to the humanity was a for-profit business. And the reason is because these nonprofit institutes and foundations, they spend most of their time actually, essentially begging for money every year, right?
Yeah,
You have to
yeah.
write proposals, and it's just scaling becomes a problem at every possible day. And so I thought, let's make it a for-profit so that we can actually scale more rapidly, have lean operations, and get some money in so that it can be self-sustaining and self-sustainable. And that's what we chose. And I don't regret that decision, but could we have gone as a nonprofit and applied for SBIRs and applied for other government grants? Probably. It's just, I think that this was the most efficient way to do it.
Mm-hmm. But it sounds like also that you were doing what we might call more traditional drug development stuff before you went this direction. What is sort of the difference between preventative medicine and traditional drug development, and how did you sort of think about that?
Yeah, I mean that's a fundamental question. So preventive medicine is figuring out why is it that nobody on a Greek island who lives to their 90s develops inflammatory bowel disease, right? Nobody. But their children who move to the city develop IBD, right? So it's not genetics. They're in the same country, same weather, similar foods, but all of a sudden their children develop all these chronic diseases. So what is it? It's not magic, it's not extraterrestrial force, it's not bad luck. It's simply there's a breakdown in some kind of a physiological pattern. And clearly, people who live the traditional way on the Greek islands, they have all the molecules in their body necessary to prevent inflammatory bowel disease, whereas their children have lost that ability and now they develop the disease. And so the concept is, can we find out what those molecules are and provide them to the children so that they never develop IBD? That's preventive medicine. Reactive medicine, which we have today, is, well, let's wait until that person develops IBD. And now let's basically find a place in their body where we can sort of stop that from happening with all the other consequences that come with it. And so, for example, for IBD, that I chose, there are pharmaceuticals that can treat it, but they're not super successful, and they
Mm-hmm.
have very heavy toll in terms of side effects and financial toll. And so, why not just figure out what is it that keeps you healthy instead of react to a disease?
Mm-hmm. And when you say that, so in this example of IBD for people from the Greek islands, it's not genetic because you're talking about one generation of difference here. You start seeing it in the children of people that don't have it. the genetics are not substantially different. They're living in the same climate. They're living in at least a highly similar culture and eating, if not identical, very similar foods, but they're missing molecules, you said, that prevent IBD. How do we know they're missing them? And if everything is very similar from one generation to the next, what's actually missing? Is it something in the
Yeah.
food or what?
Yeah. So that is really the question, what is missing? And what is going to be missing is probably many different molecules, not just one or two. Any chronic disease is going to be very complex. And here's where I think it's very important for people to understand how resilient our physiology is. Think of the human body, and we can sort of follow this thread throughout the conversation. Think of the human body as a very complex machine, such as an airline or like an Airbus A380. And that Airbus airplane has a lot of different systems that enable it to fly very fast, very long distances, but it also has many redundant systems so
Mm-hmm.
that if one screw falls out somewhere, the plane doesn't crash, right? It has multiple redundant systems. And so you as a passenger sitting on a plane, flying over the North Atlantic, if a screw falls somewhere or some electronic system fails, you're not going to notice anything. There's not going to be any symptoms. And the reason is that all these redundant symptoms pick up. But when that plane lands, if you don't fix the problem, you just keep flying, eventually enough of these redundant systems will break, and then you'll have a symptom of a plane crash. That's a very good metaphor for the human body, where we have redundant systems for maintaining our physiological health, but over time they start to break, and those are pre-symptomatic. You don't get to see those with symptoms. You don't feel any difference, but we can
Mm.
measure those. But when enough of them accumulate, then you become sick. You develop a chronic disease or cancer.
Mm-hmm.
And so it's going to be very complex to figure out all of the factors that contribute to every chronic disease. So, for example, with IBD, we have identified, when I say we, the scientific community in general, not just we at Viome, we have identified both microbial and human components that either independently or often together combine in order to cause IBD. And so in this particular case, I'll give you a couple of things. And so in one particular microbiome only specific case, what was recently discovered was a bacterium. It's a Streptococcus salivarius, which is typically living in our mouth, but it can travel and live actually in the gut. It produces a peptide called niacin, and this peptide inhibits Fusobacterium nucleatum, which is very pro-inflammatory. And there are other cases where you have microorganisms that normally process bile acids that leak out of the small intestine into the large intestine. They process them into secondary bile acids, which do not cause inflammation. So bile
Mm.
acids, think of bile acids as literally detergents. They're dissolving fats in
Yep.
your food in order to help digest. And almost all of them are supposed to be reabsorbed in the small intestine back into the bloodstream. But some of them do go through and enter the colon.
Mm-hmm.
And that's okay, because normally those old people who live on an island somewhere, they still have the microbes to convert the primary bile acids into secondary bile acids and basically render them not inflammatory. But if you've lost that microbe because you've taken too many antibiotics, you've had
Mm.
diarrhea too many times, you've eaten too many preservatives, now the primary bile acids enter the colon and they're detergents. They're stripping away the surface of your colon, and now you can have inflammation. So those are all sort of microbial root causes, but there are human root causes, root causes, but human factors that contribute to IBD, where we have inflammatory pathways that can recognize the presence of pathogens where the inflammation starts. And that's what basically triggers the inflammatory process in IBD. And that process is supposed to slow down and stop at some point in time. But if the inflammatory signals from the gut microbiome continue, then the inflammation continues. And if the person has a genetic predisposition to not calm down inflammation so easily, then that inflammation can really inflame the whole gut. And so these signals are produced by the gut microbiome to start with, and they're called lipopolysaccharides. So there are chemicals produced by the gut microbiome called lipopolysaccharides. Some of them will activate our immune system, some of them will prevent that
Mm-hmm.
activation. So it's the
Mm-hmm.
balance of those two that decides, okay, my immune system is going to get activated. But in most people, the immune system activates a little bit, and then calms down and says there's nothing to see here. But in some people, it continues to be activated, and then it causes an inflammatory reaction via these receptors called TLR4 and downstream effector molecules such as IL-1 beta and so on. And so just one disease, such as IBD, is multifactorial. It includes what foods you're eating, what microbiome you have, what microbiome you're missing that you should have, and what is your genetic predisposition. That entire combination is going to determine whether you have IBD or not.
So some people might just have a genetic constitution that makes their tendency to gut inflammation a little bit higher. Other people might naturally house certain bacteria more readily than others. Other people might lose certain bacteria because they had antibiotics as a child, or because they started or stopped eating something like a processed food that contained effectively antibiotics, preservatives, that got rid of that species. And like the one example you gave is, there are factors that the microbes produce or factors that our own bodies produce that can be pro-inflammatory in a context-dependent fashion. So maybe it's pro-inflammatory when it's this primary bile acid that our body creates. But if you have the correct, quote unquote, microbe in your gut, it's then processed into something that's not inflammatory. And so all of these things interact, and it can result in IBD in certain individuals.
And this is what we know. We don't know what we don't know. In our own internal data at Viome, we have characterized all IBD patients into 4 very different groups at the molecular level. And they have some commonalities, but they have some very big differences that we do not understand biologically what that means. And so even a disease that people call IBD, which is like a uniform disease, well, if you just take just ulcerative colitis, that is one disease, because there's also Crohn's disease. So if we study just ulcerative colitis, which we have focused on, there are 4 subtypes that we don't quite understand, what all the factors are. So for each one of these diseases, it's going to be multifactorial, and the factors that play out the disease in each person are going to be different. That's the thing. So you can basically think, this is the way I think of every disease. Let's say there are tens of factors that in a population are involved in promoting that disease. But you only need a few of those in order to actually induce the start of the disease, to start the disease. And in any one person, that combination of different factors will be different. And so you have this very complex landscape that you have to manage so that you can have a true preventive medicine.
And that would imply, so basically what you're saying is, we can call one symptomatic manifestation, a disease like IBD or whatever, but you and I could present with similar symptoms and get diagnosed with IBD or something similar, and yet the pathophysiology is very different. The origins of how that got started are completely different, or largely different between you and me, which would therefore imply that the treatment that works for me is likely not going to work for you.
That's correct. And the prevention strategy is going to be very different. And so I'd like to tie this. I really like this metaphoric comparison with Airbus A380. And so there are many reasons why a complex airplane like that is down, meaning that it is unable to fly. Many reasons, right? Because there are so many systems in that airplane. And so when someone says, oh, engine 4 is disabled, right? That would be like saying this plane has IBD. But wait a minute, engine 4 is not like one block that's either working or not working. It has thousands of components in it, and any number of combinations of faulty components can lead to that engine not working, right? And so we think the way I think about it is that we have many, many different kinds of diseases, many overlap, and each one of those, again, multifactorial, and then in each person, there's different factors that can go wrong. So what we need to do is exactly what we've done for airplanes, which is understand all the parts that contribute to a healthy, functional airplane, have a yearly or quarterly testing of every single part that contributes to that healthy airplane. And then every time that we find a faulty part, we fix it immediately. We don't say, well, it's just one part, the engine is not broken, we can wait until a few more parts break down. Because if we wait for a few more parts to break down, now the plane crashes. And in that world, humanity has decided we do not tolerate planes crashing for maintenance reasons. We're not going to tolerate 400 people dying because we know how to prevent it. On the human health side, we as humanity have decided it's okay to wait until you're sick and dying because, well, that's how healthcare makes their money. And so that's the foundational sort of mindset that we have to change, is that we can in fact completely bring preventive maintenance to the human body. We just have to do it. And the main challenge has been that no one has basically designed a business model where people are willing to pay for preventive maintenance. And I
Hmm.
think that
Hmm.
that's really the key.
Yeah.
If there are now many ways in which you can reduce the risk of chronic diseases and cancers. They are very well known. But how do you get someone to not eat something that they love
Right.
is a big problem. How do you get people to exercise when they don't like to exercise? That is the main problem, I think, with preventive medicine, is
Hmm.
that people actually have to do something about it, just like what we're doing with airliners. The airliner is not going to fix itself, and it's not going to be healthy forever. There's an army of people actually doing work to maintain that. And so it takes work. So that's really the biggest challenge, I would say. And that challenge has always been because, hey, I'm not going to stop eating steak because my uncle ate steak all his life and he lived to 95. So you can't tell me that eating steak is bad, right? We have to bring data to every single individual and say, this is why you should not eat steak, because for you, steak has these micronutrients that when your microbiome processes, it causes cardiovascular disease and your risk of a heart attack is very high. Your uncle doesn't matter because he had a very different microbiome than you. So we have to bring people the data.
Yeah. I want to there's a lot of things we'll get to, but I want you to unpack more examples like you just gave where one food can be good for one person or bad for the other person. Like, so a lot of times, even not just among lay people, but among physicians and health experts of different types, people are in this trap of thinking of foods as good for you or bad for you. Broccoli is good for you, depending on who you
Yeah.
ask. Steak is good or bad for you, depending on who you ask. What are some examples where a food could be good or nutritious for one person, but not the other? And then what do we know about the biological basis for what accounts for that?
Yeah, I think that's a key aspect of our nutritional platform, which we call Nutrition 2.0. And so I encourage people to just Google Nutrition 2.0 Viome, and they can find an article that we wrote about that. But basically, that is really one of the biggest takeaways from this entire conversation. Why is there so much conflicting information about nutrition today? If you like to eat something, you can definitely find evidence that that something is good for you.
Yeah, yeah. You can
You
justify
don't like to eat
anything if
something.
you just know how to look for the
Exactly.
evidence.
And
Yeah.
if you don't like to eat something, you can definitely find evidence that that's bad for you. And so the reason for that is that the vast majority of studies, nutritional studies out there, are epidemiological. That means they are done on a population, and they're asking the wrong question. And the question is coffee good for all humans, or is coffee bad for all humans? That is the question. The real answer is that coffee is good for some humans and coffee is bad for other humans. But that's not the question they're asking. They're asking the wrong question. And if you ask the wrong question, you can never get the correct answer. And so when you ask, are eggs good for you or bad for you? You can never get the right answer. Okay, so let's talk about examples. So I'm not on the clinical nutrition side. So, you can have a 2-hour talk with our clinical nutrition team about all of the examples, but the ones
Mhm.
I've seen are, for example, um, the ones I like in particular would be something like oxalates. So oxalates are
Mm,
pretty famous plant toxins.
yep. Or
And
spinach and other things.
yeah, so oxalates are present in some legumes, in spinach a lot. Um, and so this is a controversial topic. Traditionally, again, uh, our microbiome, like our ancestral microbiome, had plenty of bacteria to feed on the oxalates that we consumed from plants. We co-evolved with bacteria. They were basically symbiotic with us, and they would be like, hey, you're consuming oxalates. You don't have a way to break them down. They can make you sick. but I can. And so those bacteria started living with us who knows how many hundreds of thousands of years ago. And so we consume plant foods where we extract good stuff and the bacteria extract some other good stuff. But now with modern life, where we are taking too many antibiotics, too many food preservatives, eating a clean life, and we should definitely talk about all those topics.
Mhm.
Now, more and more people are starting to lose their ability to process oxalates. And so, if you do not have, if your microbiome has lost the ability to process oxalates, then you should minimize those high oxalate containing foods. It doesn't mean
Mhm.
you should stop eating all the foods that have oxalates because that would be very detrimental for you, but just minimize. And I like this particular example because it applies to me. I've had a good oxalate score since we started it many years ago until about summer of 2024 when my oxalate score went from optimal to suboptimal. It wasn't bad, just
Yeah,
suboptimal.
meaning you had the ability, you had a microbiome that gave you the ability to tolerate reasonably high levels of oxalates, and then for some
Any
reason it went
level,
down.
any level, no limitation on anything. I had exactly. I had a microbiome that was very highly capable of processing oxalates in the foods, and so I had no restriction on that. And soybeans were my superfood. Black beans were my superfood. And I am mostly vegan, and so I used to eat soybeans 3 meals a day, every single day. But last summer, the test said, oh, your oxalate score is now suboptimal. And so the computers automatically said, well, let's look at all the foods. And so what they did, what the computers did, this is all algorithms, AI algorithms, and the computers know exactly how much oxalate there is in every food. Oxalate said, go ahead and reduce soybeans and black beans because they're the highest of oxalates. Our computers have every single detail about every food in terms of micronutrients. But guess what? Chickpeas and lentils are much lower in oxalates. So now move those and eat more of those, right? And so I was at first kind of sad, frustrated, devastated about this because I was so used to, I had developed basically a full menu of foods where I was dependent on tofu. But I said, hey, this is my biology. I better listen to this test. And so that's the data that made me say, well, this is potentially serious. Let's do something about it. So I just spent one weekend, literally one weekend, reading, exploring, and experimenting in the kitchen. And I essentially devised a tofu recipe from chickpeas and lentils. And so all of a sudden, after that weekend, I started making tofu not from soybeans, but from chickpeas and lentils. And not only is that now better for me because it has lower oxalate content, but these tofus actually taste better, they're environmentally better, and they're better whole foods than
Mhm.
soybean tofu that you buy at the grocery store. And we can dive into those details if
Yeah.
there's interest. But
So for this example that you're giving for yourself, were you experiencing any problems, or was this the type of thing where something changed and the test was essentially telling you you better back off the oxalates, otherwise you're going to be probably having kidney stone issues later on or something?
exactly. So no, nothing was happening to me. And so this is one of those things where you can prevent disease years or decades before it sets in because you're understanding how your physiology is changing. And no, you don't develop kidney stones overnight. They take years or decades to develop, depending on many factors.
Mhm.
But these are the kinds of test results that can warn you about that, and you can change your diet before the disease ever sets in.
Yeah, yeah. And I mean, tell me if this is true. It sounds like there was no obvious trigger for this. Like you weren't in the hospital being pumped full of antibiotics. Something just
Zero.
changed. And I would imagine that happens to a lot of people where it's not obvious how their lifestyle is changing, but something must change. And something that would otherwise fly under the radar was made apparent to you.
Yeah, I mean, exactly. So first of all, it wasn't a dramatic change. I didn't just lose all of a sudden the ability to process oxalates. It just diminished.
Hmm.
I am getting older, so that's one thing.
Yep.
And the second thing, it's a crowded space down there. There's about 1,200 species of microorganisms, and they're all trying to survive. And they're all, it's like that metaphor putting six rats in a bucket and giving them only food for four. There's always diminishing food supply. So yeah, your everything you said is exactly right. There's nothing obvious, nothing that I've done. I didn't take antibiotics or anything, and I didn't certainly kill the ability of my microbes to process. It just diminished their ability to do so.
Hmm.
And this happens with aging.
And what, so can you start to give us a sense for like the products that you offer at Viome, how do they work? What are some of the major components in terms of what's being measured and how it's being measured and how it is
Okay.
more comprehensive than, say, just getting like a blood test?
Well, uh, we're measuring a very significant part of human physiology, and I would say that it's complementary to a lot of other things.
Mm-hmm.
I have my sort of, and I can tell you what my recommendations would be for anyone. So let me just very plainly explain what we offer at Viome currently. So we offer test kits which measure stool, blood, and or saliva. I would obviously prefer that every customer measures all three of those, blood, stool, and saliva samples. And we currently look for about 100,000 different things in those, or physiological biomarkers in those three samples. And we actually detect and quantify about 24,000 of them. And so when you go get your metabolic panel done or lipid panel, you get a few biomarkers. We report 24,000 markers from every single person. That's a very deep, it's sort of like, again, going back to the Airbus A380, a metabolic panel would say this plane has four engines, it has a fuselage, it has two wings, and it has a tail. Looks good, right? Ready
Mm-hmm.
to fly. Well, not so fast, right? But our test, we're going to send out 100 people to open up every panel, to check every screw, check every connection, check every cable, check every electrical supply, check 24,000 data points to look for, are there anomalies with those? And we can dive deeper into exactly what we do, but we can infer some lipid and some metabolic biomarkers, but for the most part, they're complementary. And so we test and we measure those 24,000 markers, and then based on which ones are out of order, meaning so these are basically, think of them as biochemical reactions. And I can dive deeper what a biochemical reaction means, but it's basically processing of some micronutrient from your food into some other chemical that the microbe doesn't need anymore. It's like micro poop, basically. And during that processing, the microbes harvest the energy from that food, and that's how they survive in our intestines. And so we measure about 24,000 of those chemical reactions, and I quantify them. And so we know which ones are too fast and which ones are too slow. And typically, if they're either too fast or too slow, that means that something is not right. And so we want to slow down the too fast ones, and we want to accelerate the slow ones. And how do we do that? I'll give a very, very specific example that people are probably very familiar with,
Mm-hmm.
which is, how do we make beer?
Okay.
We make beer by providing the substrate for a microorganism. So the substrate is the sugars that are found in barley.
Mm-hmm.
The microorganism is the brewer's yeast, or Saccharomyces cerevisiae, and the product is ethanol. And so if you want to make beer, you have to have barley and yeast. If you put those same yeast on lettuce, you're not getting your beer.
Mm-hmm.
All right. If you put that same yeast on a piece of steak, you're not getting your beer.
Yep. Specific substrates facilitate the growth of specific microbes, which is why our diets affect the microbiome composition.
Exactly. And the most important part is that metabolic output, the ethanol, the alcohol that the yeast produce. That ethanol in general is bad, but let's say there's a optimal amount of that ethanol that's produced. Let's actually switch from ethanol to butyrate, because that's actually physiologically relevant. But think of, there's a bacterium in your gut microbiome that when it consumes a very specific polysaccharide, it will produce butyrate. And if you have the right amount of butyrate, it is great. It is feeding your intestinal cells. It is providing all kinds of signals for your immune system. It is a very healthy product at the right level. If your bacteria producing too little of it, that's not good for you. If they're producing too much of it, it's also not good for you.
Yeah,
And
I would
so,
imagine that's probably a pretty general principle, right? Like the
yeah,
many molecules in the body you want at some Goldilocks level.
exactly. That's exactly what it is. It's just like cholesterol. Yeah, too high of a cholesterol is bad, but too low of a cholesterol is also as bad. And sugar Too high blood sugar is bad. Too low of a blood sugar, you're dead.
Hmm.
So even these things that we think of as, ooh, we got to get it lower and lower, that's still lower within the healthy range, but there is too low.
Hmm.
And
Yeah, and that
so
seems like an important thing too, because a lot of medicine, maybe inadvertently to some extent, but there's a lot of focus out there on lowering the so-called bad markers rather than thinking about the optimal range.
optimizing them, yeah, exactly. And we're trying to optimize everything. Okay, so let's go back to our example. You have a microorganism that can process a specific polysaccharide that you consume and produce butyrate for you.
Hmm.
You actually, most people have many such bacteria, many different species, and they consume different polysaccharides and they produce butyrate at different rates.
Hmm.
And so when we quantify your butyrate production, we know what level is healthy.
Hmm. So you're not just
And
measuring if people have butyrate or not, you're measuring how much and how quickly it's being made.
we measure the biochemical activities of the microbes for production of butyrate.
Hmm.
And so we don't just say you have Faecalibacterium prausnitzii, which is capable of producing butyrate, but it doesn't have to produce butyrate, meaning that the presence of a microorganism doesn't say anything about its biochemical activity.
Hmm.
Because, like I said, if you put brewer's yeast on a piece of lettuce, you're not making beer.
Hmm.
And so we're measuring exactly the biochemical activity for a specific chemical reaction for about tens of thousands of those. And so now, if you have just the right amount of butyrate, that's great. But if your butyrate is produced too much, then you need to reduce the production. And how are you going to do that? You're just going to reduce the substrate that your bacteria are using to produce it.
So that would
Your
eat less of certain foods that feed that bacteria.
Exactly. So the computer would automatically say, okay, you have these bacteria that are hyperactive in producing butyrate. We need to find what substrates they're using for butyrate, and let's put those foods into minimize or avoid, depending on how big the problem is, right? And if you have too little butyrate production, which is by far the most common problem in the
Hmm.
United States, in developing countries, because we don't consume fiber or enough fiber, then the computer would say, okay, this person has these bacteria and they're or whatever microorganisms they are, and they've activated these butyrate pathways, but they're starved. They're not reaching their full potential. So now we need to stimulate those pathways. And how are you going to stimulate them? You're going to provide them with the substrate.
Hmm. And
And
how do
so,
we? So if it's more common to have too little butyrate, and butyrate, those bacteria are fed by fiber. What are the consequences of having too little butyrate? What are the symptoms?
well, there are many symptoms. So some of the symptoms some of the things that I really like to bring up are butyrate literally is the food for our intestinal epithelial cells. So the one cell layer that's lining our intestines, they're called intestinal epithelium. Those cells have evolved over millions of years to actually use butyrate, that is basically micro poop from bacteria, as food. So they're using it, not glucose, but they're also using glucose, but they're using that butyrate. And so to maintain health of the intestinal lining, you need butyrate. To me, that's probably one of the best, healthy uses of butyrate, but there's many others. The one that I really, really love that I mention a lot is we all know people who get sick once or they get vaccinated once and they don't get sick again.
Hmm.
And we know people who repeatedly get sick, whether they get sick from the pathogen whether they get vaccinated or not, right?
Hmm.
And this phenomenon has been known since the advent of vaccines and since we've known about infectious diseases, that some people respond to vaccines and some people do not. And some people get sick once with something and then they're protected and others don't.
Hmm.
And just maybe 2 years ago, we teased apart, and when I say we, the scientific community, we didn't discover this at Viome. The scientific community teased apart the mechanism, and I'll now get a little technical, but basically, for viruses, the key immune cell that fights viruses is called T cell.
Hmm.
T cells will recognize human cells that are infected by the virus and kill that human cell, therefore kill the virus, because the viruses require human cells. And these T cells are cytotoxic T cells, and they're active during the infection. However, after the infection, they will die. unless they transform themselves into something called memory T cell. Memory T cells essentially live in your bloodstream for decades.
Mm-hmm.
They remember that pathogen. That's why they're called memory T cell. They remember that pathogen so that next time you get infected by that pathogen, they jump on, they kill
Mm-hmm.
it before you even have symptoms.
I see. And I would imagine in some people that process happens very readily or easily, and other people maybe not so much.
The trigger for that process is bacterially produced butyrate in the colon.
Really?
They only respond to that molecule. They don't
To
respond to
all
any
transitions from T cells to memory B cells?
to
That's a
memory
general
T
thing?
cells. Yes, yes.
Wow.
So butyrate is required for that transition. And so if you're not eating fiber towards the end of your illness or right after your illness, when you still have those cytotoxic T cells that are reactive to the pathogen, if you're not eating the right fiber for your gut microbiome, or if you have lost members of your gut microbiome that produce butyrate, then you can eat all the fiber you want. Your immune system will not remember that pathogen and you will get sick over and over and over again.
Wow.
How crazy is that?
Yeah, that's remarkable because it's like context dependence on multiple levels. Like obviously you have to get the infection for the immune system to kick in at all, and then you need to have the right diet induced microbe byproduct at the right phase of that infection to initiate the transition from the one type of T cell to the memory cell.
Yeah, it's crazy.
Wow.
So these are super important things and for our physiology, and this is just one of thousands of examples where microbiome byproducts influence our mental, physical, cognitive, and immune health.
Yeah. So talk a little bit more about essentially, so you're not measuring just the microbiome population. You're not measuring which species are present and absent. You're doing that, but you're also measuring what we can just think of as microbial behavior, what they're doing, whether or not they're producing certain metabolites or others, and how quickly or how readily they're
How
doing this.
much? How much of that? Because the quantity is very important. And
Mm-hmm.
our method is natively quantitative. And as we discussed earlier, you need to have just the right amount. You can't have too much or too little of almost anything.
Mm-hmm.
When we talk, here's another example: hydrogen sulfide gas. Hydrogen sulfide, many people say, oh, that's bad. That gives you bad gas. And there is definitely scientific literature showing that it causes inflammation, right? Too much sulfide gas. However, people who don't have enough of it, they don't have normal functioning gut. It's involved in bowel movements and in normal healthy gut. And so you have to have just the right amount. So you can't just stop eating anything that has sulfur in it and think you're going to be healthy. So you
What are
need
examples of foods that have that?
so cruciferous vegetables are best known for having sulfur. And so if you have too much, our computers are probably going to put many of the cruciferous vegetables in the minimize or avoid list. If
Mm.
you don't have enough, the computers are going to say, hey, you need to eat a lot of these.
So that would be
Or
like broccoli, cauliflower, stuff like that.
exactly, Brussels sprouts, cabbage, exactly. Those are all sulfur containing foods. And then you have specific sulfide containing molecules as ingredients for sulforaphane, I think is what it's called, that have biological activity beyond just hydrogen sulfide gas. And so, uh, going back to your question about, people think steak is a steak and broccoli is a broccoli. The way to think about food is that a typical omnivorous diet has about 15,000 different chemicals that we introduce in our body. So after you chew up all the food, like if you take all your diet that you consumed over the previous month and you blend everything and you measure all the chemicals in there, there's going to be about 15,000 chemicals. Some of those are going to get absorbed directly into our body. We do not need the microbes to help us with that. Some of them will get processed by the microbes, and they will then convert those foods into secondary metabolites, what I called micropoop earlier, what we discussed as ethanol and butyrate. And those secondary metabolites, humans have evolved to depend on for health. And it's this flood of chemicals that comes in with the food that interacts with our body and the microbiome that determines, are we healthy or are we not healthy? So it's really that big combination.
Can you walk us through? So walk us through like one of your major products that's giving like the comprehensive picture that's measuring all the different samples that you guys take. Walk us through an example of how this works, just the mechanics of how it works for someone, but then
Okay.
maybe do it. Can you give us sort of like a case study of a typical result that's very common that you see across a lot of people in terms of, something they're deficient in or something they need to cut back on or whatever?
I'll give you some examples, but there is no typical. We have 500,000 customers and we have 500,000 diets.
Mm-hmm.
There is no typical anything. So a customer would basically order a kit. And so the one I always recommend is the food, the full body intelligence, the FBI kit. They would receive a box in their home. The box has 3 sample collection kits for stool, blood, and saliva.
Mm-hmm.
Even the blood is collected at home. Of course, saliva and stool are collected at home. So they would collect those 3 samples. I can collect all 3 of those samples in under 10 minutes. Now, I'm the guy who developed this technology, so I've done this hundreds of times, so I'm pretty quick, but it's a pretty quick process. And then they ship these 3 samples in a prepaid mailer directly back to our labs, which is where I work. And we take those samples and we perform the exact same analysis on all 3 samples. And that is, we basically purify RNA molecules from all 3 samples. And those RNA molecules come from every type of microorganism you can imagine and all human cells that are in that sample. And we analyze all RNAs, independent of their source, whether they're coding for a protein or not. And all of those data get uploaded into the cloud. And then we output bioinformatically at the strain level, which microorganisms are found and at what abundance. And then what are the genes that the microorganisms are expressing and what functions are they performing? And then for human genes, what is the gene expression level for human genes? All of that bioinformatic information is now used and combined with the questionnaire that we ask our customer. And so our customers will say, these are the symptoms I currently have. So they may have IBS constipation or IBS diarrhea, or they may have depression, or they may have inflammatory bowel disease, or nothing. And then we ask our customer, what is your number one health goal?
Mm-hmm.
And that can be to reduce depression, that can be to improve lean muscle mass, that can be to lose weight, whatever they say that is. And then we ask our customers to tell us, do they have any food sensitivities or allergies? And they want to avoid any foods for any reason. And those inputs now, combined with all the molecular data the computers take, and their job is to output all foods into 4 categories, to group all foods into 4 categories. The 4 categories are avoid, means you should never eat them. And then 3 other categories that you should eat, and they're minimize, enjoy, and superfoods based on the amount that you should eat.
Mm-hmm. And
So
superfood, I imagine superfood doesn't mean it's intrinsically a superfood as that term is often used, but it means for you should eat
only
quite
for
a bit
you.
of it.
Exactly. Only for you.
So you're basically doing, it sounds like the essence of what you're doing is you're collecting 3 different types of samples and you're doing mass scale transcriptomics, gene expression measurements on all of that.
Yes, exactly. Gene expression analysis. Because we want to look at the activation of the genes, the activity, the biochemical functions, and not just who is there, because just the fact that something is there, you have no idea what it's doing. So there is no other functional gut microbiome test on the market today. They're only telling you who's there, but you have no idea what they're doing. And that's really, I knew this back in 2010 when I sort of created this platform. I was a senior scientist at a genomic facility, and we used 16S analysis, metagenomics analyses, and I knew that that's the fundamental deficiency is just because a microbe is found somewhere, you don't know which of the 4,000 genes that it's encoding are actually functional and to what level. You don't know the behavior.
Yeah, it's like what would an analogy be? Like if you had 2 cities and they both have 5 million people in them, and they might have the exact same demographic composition, but they could have very different economies and cultures because of the way people are behaving and interacting in each place.
Very different, exactly. Very different behavior, exactly. And so that's what took 6 years to develop this technology before we started Viome, because I wanted to have the right data set, the relevant data set for human health. I didn't want to just look around and say, oh, 16S is cheap, it's easily done, it's, there's kits for that, let's just start a company. I spent 6 very long years begging for money and doing research to develop this technology. Okay, so now the customer basically receives these food recommendations. They also receive supplement recommendations, which is which supplements they need and in what amounts. That's what we call results and recommendations. The results include about 70 different molecular scores. These are very much consumerized at this moment, meaning the names are consumerized. but the science under them is 100% science and robust. And we now publish 2 papers on that. So people can actually open the hood and understand exactly what we measured, how we computed everything. Everything is mathematically explained down to the last 4 decimal place for everything.
Full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.